Aloha kako. Thank you for joining us. Welcome to Line in the Sand. We're going to have four programs about invasive species, and this time we're going to be talking about a disease that's hitting our Ohia lehua forest on the Big Island. We've called it rapid Ohia death, and I have four panelists with me today to talk about this. I'm Christy Martin. I'm going to be your host for today and moderator. We have with us a forest ecologist from the Big Island, Dr. Flint Hughes. He's with the U.S. Forest Service, Institute of Pacific Islands Forestry. Welcome, Flint.
Oh, thank you, Christy.
We have Dr. J.B. Friday, who is an extension forester with the University of Hawaii College of Tropical Agriculture and Human Resources out of the University of Hawaii Manoa, but he's based in Hilo. Dr. J.B. Friday.
Thank you.
Thanks, J.B. We have Dr. Lisa Keith with the USDA Agriculture Research Service, USDA ARS. Dr. Keith?
Hi,
Lisa?
Christy.
Thank you. And we have Dr. Neil Reimer with the Hawaii Department of Agriculture Plant Industry Division.
Thank you, Christy.
All right. Mahalo to all of you for being here. I know it's a little bit out of your time to come and travel to Oahu to film this, but I'm sure everybody across the state will appreciate this information. So I'd like to set the stage a little bit for what we're going to talk about. This disease is hitting the Ohia forest, and to really get a sense of what that means, I'd like to ask Flint to talk a little bit about the Ohia and the value, what role it plays in Hawaii.
Yes, thanks, Christy. I think the importance of Ohia Metrosideros polymorpha cannot be overstated, and that's why we're so concerned about this disease. Ohia is our most widespread, dominant native tree. It's present in most of our native forests, and it's dominant in most of our native forests. It's unique in that having arrived in Hawaii millions of years ago and diversified across the various environments and habitats of Hawaii, it now occupies such a wide diversity of ecological niches that it's quite astounding. Ohia is present in lowland wet forests along the windward coasts up into the montane cooler forests, and then thinking about over into the dry forest and down even to the coast of most of the main islands. The aerial extent of Ohia across the islands, basically Ohia occupies about a 250,000 hectares, so that's approximately 600 plus acres of land on the Big Island, and about 100,000 hectares or 200 plus acres across.
Now, I'm sorry,
Yes.
I think 600 acres or 600,000?
Oh, I'm sorry,
acres.
600,000
Yeah.
acres, pardon me, across the Big Island, and then again 100,000 or so hectares or 220 plus hectares, 1,000 hectares across the remaining islands. And so when we look at the distribution of Ohia across all the islands, it's also important to note that it really constitutes our watersheds, our forested watersheds, where we get our clean and abundant drinking water for the entire state. On any of the islands you look where we're receiving our water, it's our Ohia forest. So we depend on those forests for services. Innumerable other native species, plants, birds, mammals, insects depend upon Ohia forests for habitat, for their existence. And so a decline in our Ohia forest will really have a wide-ranging and cascading effect on all of us and all of the creatures that depend on those forests.
So we actually have a really nice map that the Forest Service did available, and it shows in the blue some of the extent of the solid ohia forest, I believe, across the state on each island. But there's some other areas where perhaps it's mixed ohia and non-native tree forests.
Right. The distribution of ohia in some places it's dominant. It constitutes 80+% of the native forest. But in addition to that, if we think about all of the trees, for instance, across Hawaii Island, it accounts for 50% of all of the trees across that island, and roughly the same for the other islands. So again, that really emphasizes just how important ohia trees are to our native forest and to our islands as a whole.
Mm-hmm. So, for those of us that love going in the forest and going and seeing ohia, can you characterize going into a wet forest and seeing ohia? What does that ohia tree look like?
So, really, when I go into ohia forests, I feel like I'm entering kind of a cathedral, really, and where the ohia trees, where they're mature, they really constitute the pillars holding up the rest of the ecosystem around them. They certainly form the foundation with their roots and the litter that they create for all the organisms that are living with them. And they tend to form the dominant canopy above. So when you're in an ohia forest, you get the feeling that they really are the superstructure for that forest, both in terms of the composition of the forest, the structure of the forest, and the way that forest functions. And we also know that ohia, as a pioneer species on young lava flows, really creates the system that all the other native organisms end up eventually existing within. So all of that really, I think, again, emphasizes the importance of ohia to our native ecosystems. In addition, and because of all those things, its extensiveness across the islands, its dominance, the way that the importance of it in terms of forest function, Hawaiian culture really recognizes the importance of that species, and there are strong cultural ties between ohia trees, ohia forests, and native Hawaiian culture, and that really lives on to this very day.
Yeah. Well, rather obviously, I think none of us are native Hawaiian, but we definitely have been feeling that this disease is impacting us all the way to our hearts too. So thank you for characterizing the forest. When we first started hearing about this disease on ohia, we had so many questions. Those of us in the research community and policy and outreach, we all wanted to know just, what led up to this and what's sort of the history, what exactly are we dealing with? And so to tell that little historical part, I'm going to ask JB to go back in time a little bit. His job as an extension forester is really to be that person that anybody from the community can call and ask about, well, you're a forester, so they're asking about trees and things like that. So if you can tell us a little history about when you first started getting calls about this.
Sure, and as you say, I work with private forest landowners across the state. I'm based in Hilo, so I get a lot of calls just from our district in Hilo. People do call if they notice a problem with their forest health, with trees dying, and ohia is susceptible to a lot of, there are other diseases, very susceptible to injuries. If you damage the roots of an ohia by land clearing something, it's easy to kill it. So I remember in 2010 getting a call about some ohia are dying, and I visited the landowner. It was down in Lower Puna. And I was expecting to see a lot of places where he had damaged the trees, and these are off in the forest, and I couldn't see any particular reason. A few calls later in 2011, 2012, we took samples from these areas and found no unusual diseases. Now, a tree will have all kinds of pathogens associated with it, just like if they took a swab, they could find some germs in you, but it's not causing a disease. So we didn't know why we were seeing these trees dying when we didn't recover any kind of unusual pathogen that should be killing healthy trees. 2012, we started getting more complaints. In 2013, we started hearing from landowners that we'd go out and visit them and say, this tree was really healthy 2 weeks ago, and it died all of a sudden. And we would see these trees, the whole canopy would turn brown at once. And this, we realized this was something new, took more samples again, turned up nothing unusual in terms of what the pathogen was.
So
we did a
when
road.
you take samples, what does that mean?
What we were originally doing, because when we saw the whole canopy turning brown, we thought something must be killing the roots. Usually when a tree's leaves all turn brown, it means it's not getting any water. We thought, well, something must be killing it. So we're taking root
happens
samples.
with my plants.
It is. And again, taking root samples in Puna means chipping out things out of the rock, lava rock, with an O bar. But yes, we did some of that. We tried digging. Digging isn't very easy down there, the lava lands. So nothing unusual in that. 2013, we did a roadside survey where we just basically covered all the roads that would go through various kinds of ohia forests in Lower Puna. And we found many spots. We actually did an aerial survey of patches of trees dying back. And these would be 1 to 100 acres. And in these patches, there'd be a lot of dying trees. Not all the trees together, so it wasn't a typical root pathogen that would be spreading out like mold on a bread from a given spot. It was just an area with a whole lot of trees dying. And then there would be healthy forest for a while, an area of more trees dying again. It wasn't until 2014 that our diagnostic lab at UH with Brian Bush identified fungus ceratocystis. Now, it's a well-known pathogen, but it was never before described on ohia, because we have databases going back decades actually on what disease organisms exist on what trees or what plants. Ceratocystis had never been identified on ohia. That's when that was really the first clue. At that point, we brought in Dr. Lisa Keith with USDA ARS and started really examining what's going on with that. She conducted the laboratory and greenhouse studies to identify that as a new and unique disease-causing organism that was associated with this mortality we were seeing. So on a landscape scale, we went back to that spot that I visited in 2010, and sure enough, we found the fungus there. So that's the first time I think that I saw disease on the landscape being caused by this. Now, obviously, it was there earlier. And when it started going from 1 tree to 2 trees to 4 trees, was obvious some years before that. By 2010, it was enough that landowners were starting to notice. By 2013, driving around, we found a lot of spots on it. 2014, we found many locations of the disease, mostly below Pahoa and Lower Puna, stretching up to the Puna subdivisions towards Keau. But then by 2015, we found it spreading down through Kau and up the Kona coast. And that's kind of the distribution where we've seen it today. We've not recovered any disease north of Hilo, so we have seen it in the Wailuku River watershed. We have not seen any disease in the Hamakua, North Hilo, or Kohala districts yet.
So the Wailuku watershed is what feeds, is that what feeds Akaka Falls, or where does that come out?
The Wailuku River comes out right in Hilo. If you leave Hilo across the Singing Bridge, that's the Wailuku River. So the watershed is the whole drainage area above that. Sadly, we've seen the disease all the way up to the very top of the ohia forest as you drive up Saddle Road there. And so we've seen it there, but where we haven't seen it is up like above Akaka Falls in that watershed, and anything further north than that, we haven't seen it. We have not seen it in any other of the islands. We've gotten dozens of samples of trees that, from a distance, look like the same symptoms. When we've sampled them, taken them to the laboratory, we have not found this fungus on any of the other islands yet.
So, I'm sorry, Flint, you had something.
We had hoped that when we first were characterizing the distribution of the disease and finding it primarily in Puna with some outbreaks in the Hilo area, we had hoped that we would see some environmental constraint to the spread of the fungus and the impact of the disease in terms of killing ohia trees. We were disappointed to find, as JB had mentioned, to find the disease killing trees at the virtual top of the ohia tree line. in the Wailuku watershed. We were hoping that the fungus would be restricted to those kind of warmer, wetter areas, and that certainly was not the case. Has not proven to be constrained to that point, and we're consistently seeing outbreaks beyond that core Puna district area, both upward into montane, moist, cooler forest, and also down and around into the leeward forest where it's much drier. And again, we had hoped that the fungus would not be as virulent over on that side where it is drier. So basically what we've seen recently is that the fungus is pretty much unconstrained by environmental characteristics or environmental factors across the Big Island, that wherever ohia is growing, if that fungus gets there, if the fungus can get to that spot, it will kill those trees.
You know, I've been talking with a lot of people about this disease lately, and you know, one of the questions I think that all of us have been hearing is, well, what took you so long to discover it? And you know, from what you indicate, it sounds like you really can't find what you're looking for unless you know what you're looking for in some cases. And this is a little bit like a needle in a haystack, really.
Well, a tree is a big thing, and there are going to be a lot of different organisms on it. But the other thing to remember is there are a lot of things that kill ohia trees. There's a history of a century of work into forest health, and this issue caused this problem, this caused this problem. Certainly in 2010, 2011, there was not a level of mortality in the forest that was out of what normally the noise was of what's going on. It wasn't really until it ramped up into about 2013 that we realized something new was going on. And then 2014, we identified the disease. And again, this wasn't like dengue, where we know very well what dengue is. This was a new disease that had never been discovered before, new to science. So I think we actually turned it around very rapidly.
And that's really was attributable to Lisa, Dr. Keith, coming on board, thinking and considering the different aspects of potential causal agents, and really zeroing in on Ceratocystis fimbriata, and in very short order proving that yes, indeed, that is the primary pathogen of the disease.
We're going to take a short break here, and when we come back, we're going to talk with Dr. Lisa Keith and the third member of what we're calling the ROD Squad, Rapid Ohia Death Squad, for folks that remember the MOD Squad. We're going to talk to Lisa and find out about her detective work to really identify what this disease is. Stay with us. Thank you for staying with us. You're watching Line in the Sand. Today we're learning about rapid ohia death, a disease that is killing ohia trees on the Big Island. And I have some panelists that are helping us understand the issue. Right now we're going to hear from Dr. Lisa Keith from the Agricultural Research Service on the Big Island. She's going to talk a little bit about the detective work that she did to really help identify what was killing these trees. So Lisa, if you could just tell us a little bit about when you got involved with JB and Flint, when they called you, what was your thought process moving forward from there?
Well, as a plant pathologist with Ag Research Service, we get the same kind of calls on things like rambutan, longan, or macadamia nut. So the process is really the same, trying to uncover a mystery of what is killing a host, and in this case, ohia trees. You have to really figure out from the start, is it something biological, meaning like a fungus or a bacteria, but in ohia, the fungus? or is it something attributed to something that has nothing to do with a microorganism? So hearing the initial clues in the early part of 2014 of finding ceratocystis, which is typically an agricultural problem around the world or plantation forestry, but is a known tree killer in parts of the world, kind of the clue, the light bulb went off, maybe this is actually a good place to start. So when you're looking to see if it is a new disease and a new organism, you have to pay close attention to the typical symptoms you're seeing and always find that same pathogen. So that's kind of step one in proving the pathogenicity of an organism. So collectively, we started really doing tree autopsies, kind of like a CSI or Scooby-Doo Mystery Incorporated, getting lots of samples, paying attention to what the public's been saying. The trees seem healthy, and then a couple weeks it seems like it completely dies. So JB and Flint getting the calls from the public. These are suspect trees. We would now go and do mass sampling of it, characterizing and documenting what we see. And as soon as we started doing a lot of sampling, we could then find internal discoloration, which was really key to distinguishing a fungal problem, this new pathogen, versus drought. Okay, so yes, the canopy rapidly dies. The trees remain attached to the canopy. The leaves are there looking yellow to brown. Outwardly, maybe it looks like drought, but as soon as you start sampling inside, you see the internal discoloration, which is the presence of the fungus. So a lot of times it looks like a typical starburst pattern, really, in the vascular system, in the wood. And if you take the outer bark off, you'll actually see this darkened streaking. And again, that's known as a sign of the fungus, this vascular discoloration. So over a process of, say, the first couple months, we started just doing a lot of sampling, typical symptoms, and what we would isolate from these symptoms. And we would then start building up this population of this new fungus, ceratocystis, causing disease for ohia. At this time, we just knew really as this rapid ohia death, because that's what everybody was seeing. From that stage, it's kind of set the stage for what plant pathologists do to prove that this organism is causing disease in the plants, okay? So there's really 4 big steps that you always see the same symptoms and you're always retrieving the same fungus, okay? So that was the first stage set. Then you have to prove pathogenicity, and you have to recreate these symptoms by doing work in a greenhouse or a growth chamber. So you take pure cultures of the fungus, this new fungus ceratocystis, which in the meantime we've identified using a microscope, looking at its spore types, using more fancier techniques like molecular biology where you can match up its DNA to other things. And now we had this idea that it is Ceratocystis fimbriata, again, a known tree killer around the world. So you take the pure culture of your organism, the fungus, and you actually inoculate healthy seedlings. And you sit and wait, and what you're hoping to prove by showing this is the organism causing disease, you'll see exactly what you're seeing in the field. And that's what happened. It took some time. We had to be patient. And actually in the greenhouse, a couple months passed before anything was really observed. But then we saw the first branch wilt, and within a matter of days from that initial symptom, the whole branch died. And then within no time, essentially, the whole seedling died. Okay, so now we're pretty confident, and you do that numerous times. And you also watch a control plant, so you're doing the same thing on inoculating it, but you just put water as a control. Okay, so the process of wounding or whatever you're doing doesn't cause a problem. You don't recreate anything. But adding the pure culture of the fungus, seeing this, well, rapid, but it took some time for the fungus and, to kind of, unfortunately, work its magic inside the tree to cut off the water supply. And then when we did the same kind of autopsy on the seedlings, we saw that same internal discoloration, the starburst pattern. Okay, so again, matching up exactly what you're seeing in the forest to what now you're seeing in the laboratory. And the last step is actually then recovering the fungus you put on there, okay? And now it matched exactly what you put on. Okay, so you're only introducing one new thing, and that then killed the seedling, and so now you've proved pathogenicity. So that was really the first stage, and I mean, that took some time to do because you have to be confident the trees you're looking at in the forest is dying of the same thing with those same internal symptoms.
When you say it took some time, though, I mean, are you talking weeks or are you talking months? About how long did it take from when you got involved to saying, aha, those seedlings died?
It was a few months, actually, to be able to accumulate enough of the fungi, the same organism, and also do the experiments in the greenhouse. And again, it seems rapid, this death, but it takes some time to kind of work, and you're not seeing what's happening, but in actuality, the plant's water system is being choked off because of the fungus. But I think in the grand scheme of things, it was actually very fast working in the laboratory. So I'd say within less than half a year, we were able to do this mass sampling in the field, actually find the organism, prove its pathogenicity, and actually through peer-reviewed journal, have it become known as a newly identified disease. Yeah, rapid ohia death.
Yeah, that paper was published, I think it came out at the end of 2014. And then when we started hearing about it, just the conservation community across the state, it was really at the spring of 2015 when we talked to Flint and decided that we were going to have a meeting about it on Oahu. And so yes, I felt like it was pretty quick. So I'm always a little bit flummoxed when people say, wow, what took you so long? Like, wow, really? So I'm really impressed with all of your work together. You work very well as a team. One thing that came up a little earlier that I just want to spend a moment on, something that I think you might have said, Flint, is that, it's something that is not known to science. And then what Lisa is saying is that this pathogen, the Ceratocystis fimbriata, is a well-documented agricultural pest. Would either of you like to speak to that a little bit, maybe Lisa?
Yeah, Ceratocystis fimbriata, so the fungus-causing disease with ohia, that's the first time it's ever been documented on any kind of Metrosideros polymorpho as a new host. And you can do a lot of work in the laboratory and try to see and compare the fungus to what's around the world causing disease. And it matches 100% to nothing. Okay, so that was the challenge, that we weren't immediately able to say, oh, well, it matches exactly to coffee somewhere else, or, oh, it's sweet potato. So people have also heard Ceratocystis fimbriata is black rot of sweet potato, and yes, we have that in Hawaii for many, many years now. But the types of experiments you can do, you can make all these genetic comparisons, okay, matching DNA to DNA, and see, you can put it into a big database and try to find what its best match is. And you can also do host inoculation studies, okay? So we can isolate the sweet potato pathogen and we can inoculate Ohia and nothing happens, okay? And then do vice versa. We can take our Ohia pathogen and inoculate sweet potato and nothing happens. So Ceratocystis fimbriata is actually a large complex of fungi, which sometimes complicates things because you'll read a lot of different diseases caused by Ceratocystis fimbriata. Sometimes they have a wide host range, but sometimes
That means a lot of different types of plants? Is
Right,
that-
so it causes a disease on a wide variety of plants. But sometimes they're very specific, and oftentimes then they're renamed to even a new species, okay? So there's still a lot to learn.
Yeah.
Yeah.
Excuse me. One of the things that that we are relatively confident about is that this strain of Ceratocystis that is now killing our Ohia trees is relatively new to Hawaii. In the 70s and 80s, there were extensive investigations of looking for other pathogens of Ohia. We had concerns that Ohia was dying kind of en masse as groups across landscapes, and it turns out it was what we've called classic Ohia dieback, which has to do with the trees getting old, succumbing to stress, succumbing to some sort of disease, perhaps. The point is, during that time, scientists were looking all over Ohia forests and Ohia individuals for particular pathogens that may be causing this dieback. Never once did they find Ceratocystis fimbriata. And so we can use that as kind of a time marker to say, okay, we're confident that Ceratocystis was not here and certainly not expressing any kind of disease in Ohia before that period of time, which was, the mid to late 80s.
Yeah. I remember learning about this Ohia dieback situation when I was in school, and so it's interesting that we have that data point to see now what that difference is, and certainly it is we are dealing with something new. And so I'd like to just ask our fourth panelist to talk a little bit about when it came to the attention of really the state and the regulatory agencies. I think that there was a remarkable reaction in trying to protect as much as we can from this new disease spreading. So Neil, if you wouldn't mind telling us a little bit about, when Department of Agriculture first heard about the disease, what was your reaction to it, and what has happened since then?
The first time I think the department officially heard about it was last summer, probably about June or so, and we had received a call, may have been from Flint, I forget exactly who it was, but people on this panel were all involved in that, expressing concern to the department that there was this new disease on Ohia causing a rapid dieback and wilting and death. We were aware that there was a disease out there on Ohia before that, and I think as the panelists have described, that's something that pops up on a lot of different plants, and but at that point I think the severity wasn't quite understood. So it was brought to our attention, and my first response, well, yes, this is a very terrible thing, but what at this point can we do about it? First of all, I needed information, or we needed information on what is the distribution of this disease currently in Hawaii. If it's widespread throughout the islands, there's not too much Department of Ag can do at that point. Through the discussions over the next couple of days, it became very clear that this was, as far as what was known at the time, was restricted just to the Big Island, although there was not a lot of information about looking at other islands at that point. So we said, okay, it's very critical at this point in order for us, one of the options we had was to put into a quarantine in place to make sure it doesn't spread, do our best at least, to make sure it doesn't spread to other islands. How do we go about that? It depends on how it's spread. So, but the first thing is figure out what is its distribution. And all the information to date was that it was restricted just to the Big Island. At that point, it was just to the Hilo side. Further surveys found it was eventually had been on the Kona side. Part of the problem is, as you're probably understanding from this panel discussion, is the disease can get into a plant and not express itself. So it can get into an ohia plant and it can express itself. It might be years later before it actually shows up. So it can be somewhere else, and then you don't know it until all of a sudden the plants start dying within a couple weeks, and they just start turning brown and dying. Well, the other concern we have, okay, if we put a quarantine in place, what do we actually quarantine? How do we stop it from moving from one location to another? So we said, okay, we could attempt to stop it from moving to other islands. It seems like a no-brainer. This is an extremely important plant, and it's a keystone plant tree within our forest. It depends on this plant. We don't want to see it dying off. But there wasn't much information on that. There was some information on this disease in other plants, Ceratocystis fimbriata, possibly a different strain. A lot of that was still being worked out at that time. So we didn't know, how does it move from one plant to another? How do ohia plants get infested? Some of the things that came up is, it can't, maybe it's possibly moved in soil. What weren't real sure. Insects may be a vector. They bore into the plant. People taking infected wood, moving it from one place to another. Well, that seems like a pretty obvious way. And that's one we can glom onto very quickly with our quarantine. The others, we said, it's going to be really difficult to put a quarantine in place without having the science backing it. However, the Board of Agriculture, we brought this to the Board of Agriculture after we got the scientists' review on what is known about the disease and how it's spread. And the decision was made, this is has such serious potential of an impact into into the Aina, into the native forests, that they need to take some kind of action at this point. And that's what they did. So they were very proactive about that, put a quarantine in place by August to stop the spread of what was known at that time as potential vectors of the disease, things that could spread the disease. Things like logs cannot move between islands. Things like
So you're talking ohia logs specifically.
ohia logs, I'm sorry, ohia logs, specifically to ohia. All the information we had at that time is, well, actually that's another story. But most of the information we had at that time is that we knew that if you move ohia plants and plant parts, that those have the potential to be infested and can spread the disease. Part of the problem is, as Dr. Keith mentioned, this disease is also found in other types of plants. Now, even though it's called the same thing, me not being a pathologist, being an entomologist, we don't view insects quite the same, but there's different varieties, there's different strains that can be very host specific to that one plant. So even though it's the same organism in ohia, it may not be affecting something else. But we knew at the time that sweet potatoes had this same pathogen in it. So our question was, do we need to put a quarantine on the movement of sweet potatoes? That's the kind of things that the science had to work out. And the Board of Ag decided at that point, this is too important to wait years for that science to come. So they put that quarantine in place and are tasking the plant quarantine branch through their review process to keep the board updated on any new science that comes into place. In fact, part of what they did with the quarantine is they said the science on the soil is a bit weak at this point. So we'll put this quarantine in place in August, but we'll leave soil off of the quarantine until the first of the year, which was January 1st, 2016. At January 1st, 2016, the science was in that soil is there's good evidence that soil is a contributing method for moving the disease. Since then, the science has got even stronger, thanks to Dr. Keith, and we've got some really good information that soil is an important vector. So based on that, the board is going to, keep well, I can't speak for the board, but I would assume our recommendation to the board is to keep soil in the quarantine on this. So I think because the science is continually evolving on this, you're going to see that the quarantine may be evolving as well. Right now the quarantine is by interim rule, which is a 1-year quarantine. And so by August, we'll have to go in with a permanent quarantine. What we're doing in the meantime is we're working with the industries that are very dependent on ohia. We have ohia loggers. Anybody wants to cut ohia logs and move them off of the Big Island, they have to be tested. If they're tested and they're free of the disease, they can move those logs. If they have the disease, they cannot move those logs off the island. Same with nurseries that are moving soil off the Big Island. Those need to be tested as well. So those nurseries have to have their soil tested to ensure that it's free of the disease. If it is, then we can give them a certificate that their nursery is free, and we periodically update that. And anybody from another island who wants plants in soil from that nursery need to get a permit, and it can only purchase it from a nursery that's certified free of this disease. So that's pretty much the status right now with this quarantine.
Yeah. Well, thank you so much for working quickly on that. And I have just a couple of questions. You know, we've talked a little bit about the disease and some of the ways that it can spread, certainly through infested materials such as infected wood from ohia and possibly even the plants. What are some of the other ways that this disease can spread, the known, and what do we know and what do we not know about it?
Christy, one of the things, because other strains of Ceratocystis cause disease in other places all around the world, we know how those things move. And so we're looking at what ours is going to be similar like and how it may move. So in terms of thinking about the scale of movement, it may just move within a stand by root grafting.
And that's when roots grow into each other.
Two roots grow
Okay.
together
Okay.
and they physically graft, so the pathogen can move from one tree to another. Now in Europe, the Ceratocystis that kills plane trees in Europe moves along these avenues
I just
by
learned
root
those
grafting.
are sycamores, people.
Sycamores. Well,
Sycamores.
plane trees are related. Yeah, actually, it's the disease from the sycamore that jumped to Europe and caused disease on their trees. So it may be moving on that scale by that. But what we see on the landscape is we'll see a new outbreak several miles, hundreds of yards or miles from the next one. One of the main things that we think it is moving is by insect frass. So when a tree dies, it becomes a magnet for wood-boring beetles. And the beetles come from all over and they dig into it. They blow out a lot of frass, which is the sawdust they make as they tunnel galleries on that. That sawdust can carry the spores of the fungus. One of the things about our strain of Ceratocystis, it has a sticky spore in the fungus. So the spores themselves don't blow in the wind, but they can be attached to something, so they can move with sawdust. So as you get a big infestation, so just yesterday, for example, I was at a landowner's where he looked like the tree had these symptoms, and we noticed the base of the tree was all covered with sawdust from the beetles chewing into it. And we cut the tree, we saw the little beetle holes in the wood. That sawdust can blow for several miles, so that's one way we think it is jumping at several miles. Now, how does it get across an island? I mean, how does it get 100 miles across the island? We think that's people moving it, people, again, not realizing what they're doing, but you see a dead tree, you want to cut it up for firewood, or maybe use it for a post for a house building or something like that. Then they move the log to the other side of the island, and that's moving the disease across the island. That's why the quarantine that Neil was talking about is so important, is to stop that, the human side of how it could move. One other way that the spores do move is they're waterborne. So this is what we were talking about in the Wailuku River, for example. Once it gets in the stream, it'll flow downstream. So that's another way, a mechanism, way it moves.
It's attached, like JB mentioned, yeah.
Yeah.
Not specifically the spore, but again, attached to that infected wood.
Right.
So that's a lot of the studies, you know, really trying to figure out the biology of this fungus and what types of spores are produced. The spore type that's very resistant to different environmental conditions. We find it in the wood of ohia, which is then what can survive, you know, high temperature, low temperature, drought, not. It just kind of hangs out and waits for the right opportunity to germinate. And studying that biology and being able to develop new techniques, more rapid molecular techniques, we can then actually say, yes, there's a good database around the world of the potentials, but what is happening specifically with our strain and finding it in the frass of wood, you know, or sawdust of felled trees, using the technology to pick it up in infected soil. Doing the types of experiments Neil mentioned about having infected soil and what happens when seedlings are planted in it, and actually seeing them die because of it. Yeah.
And there are, in terms of the spores, there not only are there asexual spores, but there are sexual spores that are able to recombine. And diversify, I know you're starting to look more into that as well, Lisa, and that the ramifications for that are a little chilling in that the spores, the organism itself could evolve to not only infect and kill ohia trees, it could evolve to kill other of our other native species as well, or agricultural crops. I don't know.
Yeah, I guess when it comes to the genetic diversity of fungi, oftentimes when an organism and its host evolve together, there is a lot of diversity in the population of the fungus, and the disease oftentimes in the native areas are not so bad. But the real problem with invasive species, the fungus moves to a new place and potentially finds a new host with the right environment. And what we tend to see in our population is it's pretty much identical to itself, which does give that clue that it came from somewhere else. It hasn't been there, decades and decades to actually start recombining with itself. But Ceratocystis can also travel around on some other plant material. Okay, there are records of it traveling around the world on nursery crops, for instance. And that is one potential, that it comes to a new area, and if a lot of strains exist and they start essentially mating, what happens? What changes can occur to have a new host be now susceptible when it wasn't before, or now that the right environment is there? Because it's always critical. As pathologists, we always talk about the disease triangle. It's a host, it's the pathogen, and it's the environment, and they all have to work together to see this new disease. But again, that's the issue with an invasive species. It's so much worse than when just the plant and pathogen evolve together. And that's really what we're seeing here with ohia.
Yeah.
One of the other components to the spread of the disease that we've learned both from the literature, and I think we're seeing it in our forest now, is the susceptibility of wounded trees. So trees that have been scarred by animals, trees, we know from the literature that when trees are trimmed with infected tools, that can spread the disease. One thing we were disappointed to see up in that upper Wailuku watershed were a lot of feral cattle moving around, scarring trees. They're actually chewing on the ohia trees and creating large wounds in the trees. Well, that's a perfect place for the fungus to enter and start growing within in the tree, and eventually enables it to kill the tree. So there is that component of damage to the trees, whether it's from animals, whether it's wind damage, that interacts with the spread of the spores across the landscape.
And for example, where we're seeing it breaking out most on Lower Puna, most of those trees are probably damaged at least a little bit by Hurricane Iselle in 2014.
Yeah.
So the wind damage could be
A
real factor.
Yeah, that's right.
So, with this interim rule and some of the things that are now quarantined, in that you can't move them from the Big Island to other islands, because of course it is still only known to be present on the Big Island. There was some interesting discussion, I think, at the Board of Agriculture meeting about just the picking of the plant parts, like the liko and the flowers for lei. And I think the reasoning for including them in the interim rule wasn't so much that those are so dangerous for spreading the disease itself. Perhaps they could be dangerous, but because people are perhaps standing in an infected area and the soil on their feet may spread it. And then where you make that snip, that's then an entry point for the disease to get in. So it's all sort of tying together a little bit. And so with this upcoming expiration for the interim rule, I guess it's going to be August. Whenever the rule went into effect is when we have to have a a permanent rule in place. Are there things that we need to do to prepare for that?
I would say continue the science that's going on, just to beef up the evidence of what can be done to slow down or stop the spread, at least stop the spread from to other islands. For example, what you mentioned is a good example. Actually, the way it came about with the plant parts was there was not a lot known about that. We didn't know what the risk was, but we knew, or we surmised, that if someone's in the field and they're picking that, it is going to wound it. And you're right, that's going to give entryway for that disease. But also, they're handling material. They're walking through the forest. All that sort of stuff is going to be picking up more spores and spreading it to other areas as well. So if we can minimize that in some way, and then moving those plant parts that are from an infested forest, if they're moving that over to another island, has the potential to spread the disease. Now, we don't know what the risk factor are. We don't know how great of a potential it is to spread it, but it just seemed intuitive to the board that that would be a problem. So I think getting more science on that to figure out, is this really a significant risk? And if it turns out, actually, picking flowers isn't really that big a deal because, wind damage is so much greater out there, maybe that's not an issue. And that can be something that can be reevaluated. You can have the science behind that reevaluated by the board, and they may make amendments to how that permanent rule is going to look. Now, I'm not saying that's the case, that's just the process of how that would work. May turn out that everything that was on there is actually very high risk and it's going to have to maintain. But I think they need to, what's important as we move forward is get the science, the more information on it, the more science to back out what the methods are, and yeah.
Well, we're going to take a quick break and we will come back in just a moment with some calls to action, what you can do to help. Stay with us. Thank you for staying with us. We are still talking about rapid Ohia death, and we want to talk just for a few minutes remaining about the things that you can do to help. First of all, you've heard from the researchers and the regulatory agency involved. I just want to say that this is a tremendous partnership. The collaboration that's gone on between everybody in the community has been amazing. And for every person that you see here, just understand that there's probably 30 other people working on this right behind them. So mahalo to everybody working on this. And this research, getting back to what's going to have to be the new normal, because from all indications, we are never getting rid of this pathogen. To get back to the new normal, we're going to have to support all of the research that's going to have to go into trying to keep Ohia safe on neighbor islands for as long as possible. So we're asking for support of funding through any sort of program, but some things that you can do at home. JB's got a list of 5 things that everybody can do. So.
One of the things we've been doing is this handouts on 5 things you could do to help avoid spreading it. We did mention not moving wood around. One of the key issues is that the spores are sticky, so anything used to cut wood, tools, axes, chainsaws to clean them. What we found is the rubbing alcohol sterilizes, disinfects. So cleaning off your tools with rubbing alcohol. I always spray the soles of my boots if I've been in an Ohia forest before going to another forest, especially before going to a fairly pristine area. Just spraying it with rubbing alcohol, brushing all the mud off first, and then spraying it to disinfect it. Likewise, the fungus isn't actually in the soil, but if in these forests where it's infected, the beetle frass landing on the soil, you drive your truck through, you drive your vehicle through it, your ATV, you're going to get a lot of mud and then frass inside or underneath your wheel wells. So using a pressure washer to clean all that stuff out first and then washing it. And lastly, as I said yesterday, I was out sampling. I didn't go visit any other Ohia forest home, and then take your clothes, wash them, wash your backpacks, whatever. So just to keep from spreading it from place to place. And especially important for visitors to the Big Island forest from neighbor islands before you go home. clean your equipment, clean your stuff.
Yeah. And when we say clean it and wash it, that means hot water, detergent.
Absolutely, absolutely. And rubbing alcohol is the disinfectant of choice for disinfecting your tools.
Yeah. And there is a website set up for people to get all of that information. It is rapidohiadeath.org. And you can also like us on Facebook. You can dislike us and like us at the same time. All of that information is posted on the Facebook page Rapid Ohia Death. You know, a couple of other things. This isn't known to exist yet on neighbor islands. So we are watching each of the forests on each of the islands. There's different groups, the Watershed Partnerships, Division of Forestry and Wildlife, the invasive species committees on each island are keeping an eye on the forest, but they need your help. If you see ohia trees that are dying rather suddenly and mysteriously, again, you can go to those sources for what it looks like. Go ahead and call 643-PEST. That's a direct dial pest report hotline number, 643-PEST, or you can contact your Island Invasive Species Committee or Watershed Partnership or your local Department of Ag or Division of Forestry and Wildlife office. A couple of other fun things that you can do to help, though, and fun being, this is kind of a sad topic, but here's a program that was set up by Lion Arboretum. Want to tell us about the seed saving program?
Sure. One of the things that Lion Arboretum has done for many years is save seeds of rare and endangered plants. However, because ohia is threatened now, they've just launched a project to save seeds from ohia across the island. So ohia is very diverse. There are many different varieties of ohia and several different species across all the islands. And they want to save seed for future propagation, future reforestation, and also to help in the search for resistant varieties of ohia. So they are doing a crowd social media fundraising campaign for that. You can go to GoFundMe Ohia Love or go to the Lion Arboretum website and learn how to donate that. So people can donate to this effort.
All right, go to Ohia Love. We have come to the end of our time at our program. I'd like to thank each of our panelists for joining us today. Thank you for your expertise and your hard work on this. You have been watching Line in the Sand. It is funded through the Hawaii Invasive Species Council, through the support of the Hawaii State Legislature. And I would also like to thank the Educational Media Center's crew from the Leeward Community College. I'm Christy Martin. Thank you for watching. Aloha.